Okay, in this video I want to talk about transgenics. We're starting off with transgenics. Okay, now inserting plant and animal cells with modified genes, right? So a plant or an animal with a new gene is then transgenic by definition, okay? Okay, so let me write that down. Inserting plant and animal cells with modified genes, okay? The plant or the animal with this new gene is called transgenic. Think of the word transition, transgenic, transition, and genic gene, okay, a gene transition. There's a transgenic species. Okay, let me now talk about controlling cancer in light of this, okay? Now, to control cancer, there's a simple way to do it. You basically switch off cancer genes. Cancer is basically an uncontrolled growth, as you know, of abnormal blood vessels. So cancer equals uncontrolled growth, okay, of abnormal blood vessels. Now, 2008, researchers at the University of, at Western Australia, at the Western Australia Institute for Medical Research, discovered a gene, okay, that can be switched off. So that could reverse the growth of blood vessels inside tumors. So, quickly mention abnormal blood vessels. 2008, researchers at WA Institute for Medical Research discovered gene that can be switched off. Okay, that basically reverses the growth of the blood vessels inside the tumors, and the hope is that one day eventually the tumor can be stopped. Which is a bit surprising because we're now into 2020 and it's been 12 years. Okay, so now let me actually get into the nitty-gritty of biotechnology. I'll talk about developing vaccines. Yeah, remember, vaccines are basically they basically control bacterial diseases, right? Such as the whooping cough or the flu vaccine, right? So vaccines control bacterial diseases, okay, like the whooping cough. And they work by causing your body to react, okay, as if it had been infected by a pathogen. And a pathogen is a disease-causing organism, right? So work by causing body to react as if being infected by a pathogen. The pathogen, remember, is a disease-causing organism. Okay, think of the flu vaccine. So I took the flu vaccine last year. It makes a big difference because now that when I get the flu, it's not as severe, right? But it's spread over a much longer time period. So instead of getting it for 3 days, I might get it for 6 or 7 days, but it's not as severe. So that's the great thing about vaccines. Now, in terms of the bacteria, right now, bacteria, okay, they've got basically they've got different proteins on their surface. Okay, so they have various proteins on surface. Okay, and some will actually cause the body to start producing antibodies. Okay, when infected by the bacterium that's attacking the body. Okay, some will cause body to start producing antibodies, okay, to combat the bacterium. Now, potentially a vaccine could be produced using any one of these proteins, right? We basically, so in terms of vaccines, okay, we need to identify the genetic code that causes the surface proteins to be produced, okay? And scientists, they analyze the surface proteins and they identify those most likely to cause the antibodies to be produced, okay? So scientists analyze, and I'll draw a picture later on to show you what's happening, and identify most likely to cause antibody production. Okay, there may be hundreds of proteins, right? And the genes for proteins are then isolated and spliced into plasmids of bacterium, right? So the genes for protein then isolated and spliced into plasmids of bacterium. Okay, this is a process. And for example, E. coli. Okay, E. coli is a bacterium that's found inside human intestines. Okay, and it can be grown easily in the lab. So E. coli, okay, they produce the proteins coded for by the splice genes. Now, how does this actually work pictorially? So let me just draw a diagram. It might make it a bit easier. So here's a bacterium. It's basically a ball. And the surface proteins are basically hanging on to the bacterium. Okay. This is a pathogenic disease causing bacterium. This thing, here are the surface proteins. And these balls, basically. Okay, now then we get genome sequencing of the bacteria, or the DNA. So genome sequencing of bacteria / DNA, right? It's made up of DNA. Genome sequencing is basically identifying different chromosomes in there, different DNA. Okay, pretend these are different colors. Okay, let's say this one corresponds to chromosome that looks like this, DNA like this. This corresponds to this. This corresponds to this. This corresponds to this. Okay, now this is basically the gene spliced into DNA of E. coli. Okay, so that's a gene spliced into the DNA. This is DNA, as you can see here. Okay, now a lot of these, okay, if there's large amounts of protein produced, there's large amounts of proteins produced.